Hello and assalamu alaikum, dear friends. So today we are going to start the fourth lecture of the series, Processes of Recombinant DNA Technology. So in this lecture we will be talking about the stages of recombinant DNA technology, preparation of recombinant DNA, insertion of recombinant DNA into host, selective markers. So let us start. The, the different stages of recombinant DNA technology include number first, isolation of genetic material. As we all know that DNA is the genetic material of almost all living organisms. So excepting few retroviruses, DNA is actually even the genetic material of even viruses. So this DNA, as we know, in case of the living organisms having cellular structure, it is located inside the cytoplasm or inside the nucleus of a eukaryotic cell. This DNA is first to be isolated from the nucleus or the cytoplasm and then taken out. So for this, the cells from where we obtain this DNA is treated with different types of enzymes so as to break open its cell wall, its cell membrane, nuclear membrane, etc. And even the all biomolecules excepting the DNA, they are also to be digested, and for that purpose the enzymes are added so that all other biomolecules are digested excepting DNA, which we want to isolate. Now for this reason, so the first step is breaking open of the limiting membranes. If it is a plant cell, then we have to break open first its cell wall. For that purpose, this plant cell is being treated by the cellulase enzyme. Cellulase enzyme will break open its cell wall. If it is a fungal cell, then fungal cell is being treated by the chitinase enzyme, because we know that fungal cell wall is formed of chitin. And if it is a prokaryotic cell, they're having a peptidoglycan type of cell wall, then it is being treated by lysozyme. And if it is an animal cell without any cell wall, or what we call as it is a naked cell, then we simply place such cell in a hypotonic solution, and this hypotonic solution, because of end osmosis, it is cell membrane will burst open, the cell will burst, releasing its contents into the solution. Now once into the solution, now the second step is the digestion of the other biomolecules. So for digestion of other biomolecules, we add different types of enzymes, we add lipases, we add carbohydrates or amylases, and we add proteases, all such enzymes which will RNases, all such enzymes which will digest all other biomolecules excepting our DNA. So now inside in this solution, the cell solution, there is now a present DNA which is settle in the solution, and now we want to precipitate it out. For that, what we do is that we add to this cell solution, to this mixture rather, ethyl alcohol, chilled ethyl alcohol rather. So that ethyl alcohol will result in precipitating out of the DNA. So ethyl alcohol or chilled ethyl alcohol, or sometimes even rubbing alcohol, which is propane to all, that actually precipitates out the DNA from the cell mixture, and we take out that DNA by the process of spooling. So this completes the first step, that is isolation of genetic material. Now the number second stage or the second step of recombinant DNA technology is the cutting of DNA at specific locations. Now DNA is to be cut at specific locations. For that reason, we use special type of enzymes which are popularly called as molecular scissors. These molecular scissors are nothing but special type of endonucleases called as restriction endonucleases. So I will write down here, these are restriction endonucleases. Restriction endonucleases. So restriction endonucleases, they are special in the sense that they cut DNA at specific locations. Those specific locations are called as restriction sites. And these restriction sites, they are very specific for a particular type of restriction endonuclease. So there are several types of restriction endonucleases that have been discovered from different types of bacteria. Some 200 types of restriction endonucleases. I'm not sure about the exact number. So they have been isolated from different types of these bacteria, and they cleave this DNA at specific sites, at specific sequences. Those sequences are palindromic. We will be discussing about them in later lectures as well. So that DNA, those restriction endonucleases will help us in cleaving DNA at specific sites. Number third stage or third step of our recombinant DNA technology is the amplification of gene of interest using PCR or polymerase chain reaction. So this again we have discussed in length in previous online lectures. You can go through that process. So PCR is the technique by which we can amplify a gene of interest, a DNA fragment, many times, billions of times in just overnight. Number fourth step, which we will be discussing in this lecture in detail, is the preparation and insertion of the recombinant DNA into host cell or organism. So we will see how this recombinant DNA is prepared and then how is it inserted into host cell. Number fifth point or number fifth stage of recombinant DNA technology is obtaining the foreign gene product. Actually the main objective of recombinant DNA technology is the production of a gene product. It can be either a vaccine, it can be either say for example a hormone, or it can be any enzyme or any other such biomolecule which is of commercial or which is of medical importance. Number final and last stage of the recombinant DNA technology is the down processing. So let us start with our preparation of the recombinant DNA, how this recombinant DNA or rDNA is prepared. Now rDNA or recombinant DNA, it is prepared from a vector DNA, which can be plasmid, and alien or the foreign DNA carrying the gene of interest. We know what is now plasmid. Plasmid is actually, there are certain prokaryotes like bacteria, they have an extra chromosomal circular molecule of DNA called as plasmid. So suppose this is a bacterial cell and this here is, it is prochromosome or what we call as nuclei, and apart from this prochromosome, or they have, some of the bacteria have extra chromosomal circular molecule of DNA which confers them special properties, and that circular molecule of DNA is called as plasmid. So here is the plasmid, this is plasmid. So this plasmid is now a DNA molecule, circular molecule of DNA, plus the DNA or the foreign alien DNA carrying the gene of interest. From these two, we prepare our recombinant DNA. So for that, this plasmid DNA and the foreign DNA from which we obtain the gene of interest, these both types of DNA are being treated by the same type of restriction endonuclease. Restriction endonucleases are of different types, and different types of restriction endonucleases, as I said earlier, they are specific, they have a specific restriction site. And it is to be remembered that while making a recombinant DNA or rDNA, we have to make use of the same type of restriction endonuclease, both in case of plasmid or vector DNA as well in case of the passenger DNA or the gene of interest. So I will be discussing this in next slide and diagrammatically as well. Suppose here this is our, pardon me, suppose here is our this plasmid DNA, this is our plasmid DNA. This plasmid DNA, as you can see here, the sign color region, here is it is restriction site, and here is it is antibiotic resistance gene. So this plasmid carries a gene which will confer the bearer of this very plasmid antibiotic resistance. So if we treat this plasmid with a restriction endonuclease, it will cut open this plasmid. So that cut open plasmid will look something like this. So this is the cut open plasmid having sticky ends. These are the sticky ends of this plasmid. The next step is that we treat the foreign DNA from which we have to collect this our gene of interest. So this is suppose a foreign DNA, and here it carries a gene of interest whose product we want to obtain. So please note that this gene of interest is being flanked by the restriction sites on either side. So if we treat this foreign DNA with the restriction endonuclease, it will produce two cuts in the DNA, in this foreign DNA, one along the left hand side and another on the right side. So once we do, we will have a gene fragment having the gene of interest in the center and flanked by the segments of the DNA on either side, like this. So here is our now DNA fragment containing gene of interest. So it is having these sticky ends, the complementary sticky ends that were produced in the plasmid. So next is that we fuse or combine the DNA fragment, this DNA fragment containing gene of interest, and the cut open plasmid, like this. So here is the cut open plasmid here, and this is our DNA fragment. These are ligated, these are fused by another enzyme which is called as DNA ligase. So ligase is an enzyme which fuses two DNA molecules. Remember, restriction endonucleases are the enzymes which cut DNA molecules. So for that reason, they are popularly called as molecular scissors. Ligase, in contrast, is the enzyme which fuses DNA molecules. So therefore, this very enzyme is popularly called as molecular glue. So once we have treated these two fragments of DNA, these two fragments of DNA, one is the gene of interest carrying DNA and another is the vector DNA, vector plasmid, what we obtain is the recombinant DNA. So here is our recombinant vector or recombinant DNA carrying the foreign gene of interest, or what we call as passenger DNA. This is a vector DNA, this plasmid, and it carries a foreign fragment, which is the passenger DNA or our gene of interest. So you can see in this diagram the whole process, how we were able to produce recombinant DNA. This is again this plasmid having the restriction site. This is the foreign DNA carrying the gene of interest. We treat both with the same type of restriction enzymes. Restriction enzymes, what they do is that they will produce complementary sticky ends. So here complementary sticky ends are being produced. Then the both are ligated by the enzyme ligase. Once we do that, what we obtain is the recombinant DNA. So this is the recombinant DNA molecule. Now once we have formed this recombinant DNA molecule, next step is to insert it into the host so that we are able to produce the end product or gene product of this foreign gene of interest, of this foreign DNA fragment. So number next is insertion of recombinant DNA into the host cell. Now for this, both direct and indirect methods are used to introduce the ligated or the recombinant DNA into host. So for among the direct methods, there are some physical methods and there are certain chemical methods. So physical methods include microinjections, and chemical methods include the electroporation. Now what is electroporation? Electroporation is the process by which temporary pores are formed in the host cell. So those temporary pores, they are being formed by certain enzymes like lysozymes. Say for example, if the host cell is a prokaryotic cell, say for example Escherichia coli, so addition of a little bit of lysozyme will produce holes in its cell wall, and through those holes this DNA, recombinant DNA, can enter into its cytoplasm. Or sometimes what we do is that we treat the host cell with calcium chloride or sometimes even calcium phosphate. That will also result in insertion of our recombinant DNA into the host cell. Next method is sometimes called as chemically mediated transfer. This chemically mediated transfer is done by a chemical called as PEG, or polyethylene glycol. So polyethylene glycol, because of treatment of host cells with polyethylene glycol, the host cells are able to pick up or take the foreign DNA or the recombinant DNA molecule. Next physical method is this biolistic gun method or gene gun method. This is an instrument wherein with the help of which we are directly able to insert the foreign DNA into the host. Next can be a indirect method, and in this indirect method, what we do is that we make use of certain viruses like bacteriophages. Bacteriophages are used to introduce this plasmid DNA into the host bacteria, or sometimes even simian viruses can be used for animals. And cauliflower mosaic virus or tobacco mosaic virus, they have also been used for transfer of these foreign DNA into the host plants. So here is a diagram of this microinjection. Here this is the microinjection. With the help of this microinjection, we are able to directly insert our gene of interest into the host cell, gene of interest or the recombinant DNA molecule into the host cell. And this is here the biolistic gun or the gene gun. So here you can see a researcher using this biolistic gun to insert a plasmid recombinant DNA probably into the host cell, into the leaves of the host directly. Now, once this recombinant DNA has been inserted into the host cell, number next is to find out whether the host cell, particularly in case of the indirect method, has taken up the DNA molecule or not, has taken up the recombinant DNA or not. So what we do for that is that if the recombinant DNA bearing gene for resistance to antibiotic ampicillin is transferred into the host cell, say for example here Escherichia coli, the host cell becomes transformed into ampicillin resistant cell. Say for example here this is the host cell, Escherichia coli cell. If we insert in it the recombinant DNA molecule, say for example, which is here marked by this red color, if this recombinant plasmid also carries antibiotic resistance gene, say for example, which is marked here is in green color, here this is an antibiotic resistance gene, say for example it confers the recipient ampicillin resistance. So this bacterial cell now will become resistant to ampicillin, which means that if we culture this cell in a culture medium having ampicillin, this type of host cell will be able to survive, or we call this type of cell as the transformed cell. See, here you can see this is the host cell containing this, having taken up the recombinant DNA molecule carrying our gene of interest as well as the gene which will confer it ampicillin resistance. So if we culture it in a nutrient medium containing ampicillin, this type of cell, this cell here, this will be able to survive. While as this cell which has not taken up the recombinant DNA molecule, therefore it will lack the ampicillin resistance gene. Therefore this type of bacterial cells will not be able to survive in the nutrient medium containing ampicillin. So this cell here is transformed cell, and this cell here which lacks this recombinant DNA, this is the non-transformed cell. So this ampicillin resistance gene here, which helped us in distinguishing the transformed cells from the non-transformed cells, this is called as selective marker. So ampicillin resistance gene in this example which we discussed just now is the selective marker. Why? Because it enabled us to identify transformants from the non-transformants. So that is all for this lecture. In next lecture we will be talking about how we are able to obtain the foreign gene product. Thank you very much.